Welcome. We discuss about the isolation of bacteria by stick plate method. There are various techniques which are used for the isolation of the bacteria as stick plate, pour plate and spread plate. Now we discuss stick plate technique for the isolation of the bacteria. Isolation, it is nothing but the separation of the pure culture from the mixed culture. Pure culture, this is the growth of a single type of the microorganism, and mixed culture, this is the growth of two or more than two microorganisms. This inoculum, it is sticked over the surface of the agar in such a way that it thins out the bacteria as the sticking increases, and after this incubation we get the formation of discrete colonies. There are various types of the sticking as 4-quadrant method, zigzag, T-shaped. Mostly the 4-quadrant method, it is used for the isolation of the bacteria. We have to occupy the total area of the agar surface while sticking, and this is not wasting the agar surface. Requirements: provided bacterial suspension, sterile nutrient agar plate, nichrome wire loop, gram stains as 1% crystal violet solution, grams iodine, 95% ethanol and 0.5% safranin. Procedure: firstly, gram stain the provided suspension to see the density and grams nature of bacteria present in the suspension. If dense suspension it is there, we have to dilute the suspension and then used for the sticking purpose, and if the suspension provided it is the dilute one, then there is no need to dilute the suspension and directly we can use for the sticking purpose. Stick on the sterile nutrient agar plate provided. Incubate the plate at 37 degrees Celsius for 24 hours. After this is the incubation period, and after this incubation period observe and note down the colony characters. The colony characters which have to be noted, these are size, shape, margin, elevation, color, opacity, consistency, gram staining and motility. Depending on the difference in colony characters, number of colonies are labeled as colony number 1 and colony number 2. If we see in gram staining before isolation as gram positive cocci and gram negative rods, then we can make a guess that in the provided suspension two types of the microorganisms are present, and after incubation they will form the two colonies, and this colony number 1 it is having the gram positive cocci, and in the colony number 2 it may have the gram negative rods. In this way, this is the isolation of the bacteria from the mixed culture. Thank you for your listening.